Hello, fellow participants in National Webinar on Avenues in Plant Sciences. Dear friends, I myself, Dr. Bhagwan Mali, here to introduce our resource person, Dr. B.B. Chaugule, who is going to deliver a talk on Astaxanthin from algae. Dear friends, Dr. B.B. Chaugule is Emeritus Professor of Bharati Vidyapeeth, Pune. He worked as a professor and head of the Department of Botany in Savirtibai Phule, Pune University. He is also a Emeritus Professor of Savirtibai Phule, Pune University. He has published more than 80 research papers in national and international reputed journals. He actively participated in national and international conferences for presentation of his research work. He is having a research experience of 48 years in various disciplines of botany such as algology, marine botany, hydrobiology. Also, he is a consultant for the industries like Gokte, Salt and Chemicals, Reliance Industries. By seeing his research work, he was awarded by Krishnamurthy Institute of Algology in 2016 by giving him Lifetime Achievement Award in Algology. He has also given a kind guidance to more than 21 students for their MPhil degree and 18 students for their PhD degree. He is having administrative experience on his credit. He has worked as a HOD of a botany department, Savirtibai Phule, Pune University from 2006 to 2009. Worked as a officiating registrar of Savirtibai Phule, Pune University in 2007. Also, he was a member of a research funding agencies such as UGC, DST, CSR, DBT. Also, he was a chairman of board of studies in Savirtibai Phule University. Also, member of a board of studies of Shivaji University, Bharati Vidyapeeth, and member of a academic council too. So, such a scientist, administrator, Dr. B.B. Chaugule, deliver his talk, Astaxanthin from algae. Thank you very much.
Hello everyone. At the outset, let me thank Principal Dr. Chandrasekhar Murumkar, Dr. Shashikant Savan, and other members of the organizing committee of the national webinar and the college authorities for giving me this opportunity. Today, I will be talking on a very precious compound derived from a species belonging to a neglected group of plant kingdom, namely algae, and the compound is astaxanthin. Astaxanthin is a naturally occurring biological pigment belonging to keratin group. It is symmetric ketocarotenoid and when present in predominance gives red to blood red coloration to the organism. Richard Kuhn, a German biochemist and Nobel Prize winner in chemistry, is credited for discovery of red pigment in 1938 from lobster shell and eggs which he named as astaxanthin. Astaxus meaning lobster, xanthin indicating to group xanthophylls to which it belongs. Astaxanthin is a lipophilic molecule, but the existence of keto and hydroxyl groups at the end causes an affinity with polar site of phospholipids of a membrane surface. It is believed that such physical property enables astaxanthin molecule to exist perpendicular in the lipid bilayer structure of membrane, and astaxanthin protects cell membrane against an oxidative attack from outside of the membrane as well. Now let us look into the sources of astaxanthin. The natural sources of astaxanthins are yeast, salmon, trout, krill, shrimp, crayfish, lobsters. The animals are unable to synthesize astaxanthin de novo, and through food chain they manage to acquire astaxanthin. However, increasing pollution in the coastal water is leading to accumulation of heavy metals and pesticides in the body of animals, making seafood unsafe for human consumption. Besides, vegetarians and lot others nowadays are moving towards vegetarianism. Therefore, astaxanthin derived from plant source is in demand. The sources of astaxanthin are yeast like Xanthophyllomyces and Raffia. However, the production of astaxanthin from yeast proved to be low yielding due to problems in down processing. The other source is petals of Adonis aestivalis, which contain astaxanthin. However, reports on Adonis induced poisoning cases in both humans and animals by Hurst in 1942, Galley et al. In 1996, and Woods et al. In 2004 resulted in considering the plant unsuitable for astaxanthin production. Synthetic astaxanthin is produced from petrochemicals via multi-step process. It is used as food colorant and additive in fish feed for pigmenting the flesh of certain commercially framed fish like salmon and trout. Although synthetic astaxanthin is cheaper compared to natural astaxanthins, but it has shown some toxic effects. Therefore, the remaining source, namely algae, are considered as only safe and suitable options for astaxanthin production. Both marine as well as freshwater, microscopic as well as macroscopic algae are in a race to serve as resource of astaxanthin. Microscopic algae like Chlamydomonas nivalis, now which is known as Chloromonas nivalis, causing a phenomena called as red snow. Then species of Chlorella zoppingensis, Chlorococcum neochloris are known to produce astaxanthin, but the best source of astaxanthin is Haematococcus, as may be seen from this table. If you look into the table, and compare the percent astaxanthin on dry weight basis. Haematococcus shows maximum content starting from 2.7 to 3.8% on dry weight basis. Uh, Macroscopic algae like Enteromorpha intestinalis, Ulva lactoca of green algae, and Catenella repens of red algae are also with astaxanthin, but but the the amount is very low, and therefore those are not considered for commercial utilization. Thus, the most suitable candidate among the algae for commercial astaxanthin production is Haematococcus, and the species of Haematococcus that is extensively used is Haematococcus pluvialis. The genus Haematococcus is a member of green algae, which was established by a German military officer turned botanist at later stage, namely Flotow, who in 1844 established genus Haematococcus. The red coloration was observed at that time, but it was thought due to hematochrome. It took almost 100 years to know the exact nature of the pigment. There are about 8 valid species of the genus, among which, as I said before, Haematococcus pluvialis is used as a source of organism for astaxanthin production. The alga is unicellular, freshwater in its distribution, motile. Motility is due to two flagella, anteriorly produced flagella. The most peculiar feature of this alga is that the protoplast and cell wall are separated, and the gap is filled by mucilage, and through this mucilage protoplasmic connections, which may be simple or branch, arise from the protoplasm and reach the cell wall. Chloroplast is cup-shaped with wavy internal margins, contain two to many pyramids. Reproduction is by aplanospores or by zoospores. In nature, alga will be seen in two stages, either in a motile or non-motile, and under unfavorable conditions it starts accumulating astaxanthin from the center, and from center both in motile as well as non-motile cells, and from the center then it spreads towards the periphery and ultimately occupy the entire cell. There are reports of Haematococcus from many parts of India, but most of the reports need to be verification, needs to be confirmed. For example, the uh, report now, the figures of the report which I am showing over here shows a pond with red colored water and a planospores on the right hand side. Now this report about which I am talking is quoted everywhere as the first report of Haematococcus from our country. If you look very carefully into the bloom, red colored bloom, as well as the planospores, raises doubt about the presence of Haematococcus. In fact, it is a bloom of Euglena. The bloom of Euglena is always nystonic, forms at the surface. The pond is much bigger than the Haematococcus that prefers, and the cysts, if you look very carefully, which will show typical euglenoid organization. Here is the typical habitat of Haematococcus. Haematococcus grows in a very small water body like pit holes in the rocks which are intermittently dried. In larger water bodies you do get Haematococcus, but it will be in a very small numbers because they cannot compete with other organisms. And thus taxonomy is also equally important uh, when you look for any product even it is not outdated. Besides, ecological knowledge is also required. What type of environment in a organism that while cultivating while undertaking cultivation of any organism or alga, knowledge of its life cycle is very much essential because this knowledge will help to grow alga into artificial medium as well as to obtain the maximum biomass and also the desired product. Haematococcus during reproduction loses its motility, undergoes division to form 2 to 32 cell structure. Each cell either germinates into non-motile coccoid cells or produce flagellated cells which swim in water. The coccoid cells again enlarge, undergo division, the cycle continues. Under unfavorable conditions, both motile as well as aplanospores start accumulating astaxanthin as I mentioned before. This accumulation starts from the center as seen in this figure and then spreads towards the biomass of Haematococcus is produced in open ponds as well as closed system. The open pond that is used is a raceway type of pond where water is shallow, water depth is maintained very shallow so that light can penetrate up to the bottom. As seen in the right side figure that the movement in water is carried by a paddle wheel. If you look into the photo which is there on the left side, again in that photo in the left side you see a green biomass very densely formed. Now when this dense biomass is formed, then the alga is exposed to stress like nutrients or temperature or light or salt so that it starts accumulating astaxanthin. Thus, it is a two-stage biomass production to obtain a desired product. For human consumption, however, Haematococcus grown in open ponds is unsafe. Therefore, under such instances, closed systems are preferred as shown in this PPT. These are photobioreactors, series of photobioreactors with facility to provide light, air and nutrients. Again here also you have a two-stage production or development. Left side, the photoreactors that are there on the left sides are green, and if you see on the extreme right side, there is a red biomass which is with full of astaxanthin into it. Now, what is the necessity to take astaxanthin or any other supplementary food? Because to carry our routine task, we need to take a full balanced diet that supports or that supplies sufficient nutrients to our body. We often need to take supplementary food as vitamins, minerals, fatty acids, polyunsaturated fatty acids like omega 3, omega 6, or astaxanthin, or carotenoids, and other phytochemicals who are essential for to fortify cellular protection. They help in repair and regeneration as well as support the renewal process. Astaxanthin activity is tremendous as far as its antioxidant ability is considered. Astaxanthin is 10 times as antioxidant activity of astaxanthin is 10 times more than beta carotene, 65 times more than vitamin C, 100 times more than alpha tocopherol, and 550 times greater than vitamin E. Thus, astaxanthin is a super antioxidant. In fact, it is known as king of antioxidants. Because of these wonderful applications, astaxanthin is used in human health and supplementary food. Some of the activities that are attributed to astaxanthin are antioxidant activity. It protects from UV rays. It has an anti-skin cancer property. It is anti-inflammatory. It has an anti-gastric activity. It is known as anti-diabetes. The one of the most important uh, use of astaxanthin is in cardiovascular prevention. It is also known to have a immune response and as well as useful in neuroprotection. In addition to human health, astaxanthin is also used in aquaculture and poultry industry. Last 20 years in aquaculture and poultry industry, synthetic astaxanthin is used because it is cheaper. But because of toxic effects, as the synthetic astaxanthin is produced from petroleum base with a series of reactions. In addition, there is an increasing preference towards the naturally produced astaxanthin. It is a very safe and effective compound for fish and skin pigmentation of the fishes. Astaxanthin is known to provide adequate growth and reproduction in commercially valuable fishes. In addition, it is known to improve health and fertility of the chicken. Also, it decreases their mortality and improves pigmentation of the eggs and egg production. If you look into the global market of the astaxanthin, there are 16 leading commercial companies spread over in countries as USA, Israel, Japan, Sweden, UK, China, Malaysia, and India. In addition, if you look into the trade, it is in billions and it is expected to rise. Very recently, in 2019, a Japanese group has come up a bioengineered Haematococcus growing in marine waters, having ability to grow very fast as well as produce astaxanthin. Now it will be worthwhile to see whether Haematococcus continue to serve as a source material for astaxanthin or it is Haematococcus that replaces. With this, I wish to conclude my talk. Thank you.